objective, that was to understand more about microorganisms and to understand about the structure and the classification of microorganisms. So let us just start with understanding what basically are microorganisms. Now, microorganisms are really small, tiny, minute organisms that are not visible to the naked eyes, but they're present in all over the environment and the surroundings. They're present in air, they're present in soil, they're present in water, they're present on the surface of your skin, they're present on any other material surface, they're present inside your body. They are basically in billions and trillions of number everywhere, and they are so minute to eyes that they are not visible unless we see them under the microscope or the electron microscopes if they are really minute. There are certain microorganisms like viruses that are really minute and cannot be detected that easily. So to observe them and to analyze them and to understand whether they are virulent actually means that do they have the ability to cause infection to the host, or how damaging can their action be on the host's body. Now you might be wondering, what is a host? So host is any organism that is subjected to the infection by the bad or the unhealthy microbes that are present around us. So in the case, I'll just explain this to you with the help of an example actually. In this example, imagine there's a little boy, he has a stomach pain occurring to him very frequently. And there's a certain bacteria that is responsible for causing this pain to him. So this ache that is created to him, so in this way he is known as a host because he was subjected to the ache by the bacteria. And in this example, the bacteria is the pathogen, that is the bad microorganism that causes the infection to the host. So let us just define what is pathogens. Now pathogens can be defined as a microorganism that has the ability to cause an infection or a disease. I'll repeat it again. A pathogen may be defined as any microorganism that has the ability or the capacity to cause infection or an disease to a host. Now I remember talking about beneficial microorganisms and the bad or the pathogenic microorganisms. So beneficial microorganisms are nothing but the good microorganisms that are helpful to the humans and animals and any other organism. They help in flourishing and nourishing the humans' flora and fauna. So beneficial or the good microorganisms may also be known as microbes in short. They are the helpful microorganisms. So I'll explain to you again with the help of an example. The example here is the example of fermentation. Now you might have observed your mother leaving a bowl full of milk with a little bit of yogurt or dahi in it to ferment over the night. That means that little dahi will be helpful in converting the whole of the milk to dahi. How does this probably happen? This happens because of the good microorganisms. And these good microorganisms, they act over the night at a very optimum temperature for them to convert this milk product to a fermented product like dahi or yogurt, or for that matter even idli batters that you have. So this is one main example of fermentation. The other example would be that you would observe in your own body. Now your intestine has a lot of microorganisms in it. These microorganisms, they are actually helpful for maintaining your regular activities of your intestine. They are helpful in making the activities like the metabolism, that means the breakdown of the food that you eat at a very faster pace. So that is the other example in relation to microorganisms being helpful for humans. So let's just move further to understand about the types of microorganisms. Now there are 6 types of microorganisms that range from bacteria. I explained to you that bacteria are of eukaryotic and prokaryotic origin. So Bacteria is the first organism. The second organism is the virus. The third organism is the algae. Algae is the nothing but the green, fleshy, very slippery mass of green color carpet that you see in rainy seasons on the floor. So that is nothing but algae. And fungi also belongs to the same class, but we'll study about their structure and their morphology when I'll be explaining in detail about this to you. The next one is the yeast cells. Now yeast cells are responsible in the making of breads, in making of the bakery products. So I'll explain about how it is beneficial and how it is not beneficial also. The last one is the protozoa. So viruses again are kind of pathogens actually. So we'll start with the bacteria, the classification of bacteria first. But before we move on to understanding, let's just have a summary of this so that you understand what microorganisms are actually in better. Now I explained to you that microorganisms are the small, tiny, minute organisms that are not visible to the naked eyes. I also explained to you that they are present in the environment, all over the environment, and in billions and trillions of counts. So they are present in air, in soil, and in water. Then I defined what was a pathogenic microorganism or a pathogen. A pathogenic microorganism or a pathogen is nothing but a microorganism that has the ability to cause an infection or a disease, right over here. Then we came down to understand about the beneficial or the good microorganisms. The good microbes are nothing but the microorganisms that are helpful to the humans and animals and other living organisms. Then we came down to understand about the types of microbes. We understood that there were 6 classifications to this. The first one was bacteria, second one was virus, the third one was algae, the fourth one is fungi, and the fifth one is yeast cells, while the last is the protozoa. Now we'll be moving on with bacteria. So let us just start by understanding about bacteria. Now, what is the size of a bacteria, or what is a bacteria in short? These are the common questions I'm sure might be running right now in your brain. So a bacteria is of the size that is larger than a virus. Now viruses, I told you in the introduction that viruses are very minute, and seeing them under a microscope is also like very difficult or practically next to impossible. So bacteria are larger than the virus and need a microscope to be observed. Though they are larger than the virus, they definitely need a microscope to be observed because they are in the end microorganisms. Now the bacteria can be of different sizes and shapes and can be clustered together in clusters or can be aggregated in the chains together. So let us just understand about the shapes and different forms of bacteria. I have drawn the whole 5 types of the bacteria, the types of shapes of bacteria over here. So we will start with the first one, that's the bacillus. Now bacillus is nothing but a bacteria that is rod shaped. The structure is right over here, I have drawn it over here. So bacillus is a bacteria that is of rod shaped. The next one is coccus. Coccus is nothing but it is a bacteria that is of spherical shape. Now why these shapes actually? A bacteria, I told you, is present on the planet Earth, in this living system, ever since the period of evolution. So it has, and I remember telling you in the introductory lecture that the bacteria has adapted and evolved itself in such forms that it can possibly live in any environment, any condition, and yet can survive and be proving to be helpful, either harmful in its own ways to the living organisms. So the third shape is the vibrio. Vibrio shape is nothing but a comma shape that's like slightly curved over here. The next one is the spirillum or the spirillus, that is of the spiral shape. It rotates around each other in the form of a helix or a spiral. You have seen the spile binding springs. These spile binding springs are spiral in shape. So this bacteria, this shape of bacteria is similar to that. The next one is spirochete. Spirochete is a much more curved and much more compacted part than the spirillum, and it looks like this. It's actually long and elongated. Now the examples of bacteria, that is the bacillus, would be the rhizobium bacteria, or would be the E. coli bacteria for that matter. E. coli is nothing but Escherichia coli, and this is a very complex name. You need not really learn all these names, but for your extra knowledge, I'll be telling you about the names of microorganisms. There are a set of microorganism names that you really need to learn, and I'll explain to you about that also. The next one is the coccus, that is the spherical shaped bacteria, and the examples for this is Staphylococcus or Streptococcus. Now coccus is the kind of bacteria that can be present in a clustered form right over here, or in the way of chains. This is the chain pattern, and this is the clustered form. Whereas the bacillus can also exist only in the form of chains, chains of bacillus, chains of coccus, cluster of coccus. You define it to be like that. The next one is the vibrio. Then vibrio, I told you again, is slightly curved or in the shape of comma. The next one is spirillus, and the example for spirillus or spirillum is Treponema. Now Treponema is that's the only example for its shape. The spirochete is a curved form. Now let's learn a little bit more about the bacteria. We've known about their shapes. Let's go up onto their body morphology and the action of it on soil and human beings. So the bacteria, and I remember explaining to you in the cell structure that there is a centralized nucleus in animal cells, and plant cells the nucleus is present on the sides towards the cell wall. But nucleus is the main organelle from where the whole activities of the cells are performed. But in bacteria, there are only 3 chromosomes and there is no nucleus. Instead of nucleus, for the cell, for the bacterial cell to perform its functions, it has 3 chromosomes in it. Now 3 chromosomes. Chromosomes, what do you mean by that? Chromosomes are nothing but when DNA combines together to form a long strand, and this strand, there are a number of strands to formed with each other, and these strands, they are in the shape of helix, and they are long and suspended in the whole of the cytoplasm. So in this case, they are the free chromosomes. Then the organelles like cell wall, the cell membrane over here, and the other is the cytoplasm. These 3 are only the organelles present in a bacterial cell. And now, since we can also, I explained to you about eukaryotes and prokaryotes. So this kind of bacteria can be considered to be a prokaryotic bacteria because it does not have a nucleus, a well-defined nucleus, and all the material of it is suspended in the cytoplasm. So then I repeat the sentence again. It has 3 chromosomes instead of the nucleus, and it has organelles like the cell wall, the cell membrane, and the cytoplasm, and in the cytoplasm are the suspended free chromosomes. Now why the cell wall? Because this is one defense mechanism that it uses for itself. So the next point is we'll learn about a little bit more about the types of bacteria that are present. So the first type of bacteria is the rhizobium bacteria. Now these rhizobium bacteria, I told you they are of the bacillus shape. Now let's understand about their functions. Their functions are that they help fix nitrogen into the leguminous plants, and this fixation is carried out in soil. So Rhizobium bacteria is a nitrogen fixing bacteria found in the roots of leguminous plants or legumes. Leguminous plants like peas. So that's one example of leguminous plants. And how does it really fix nitrogen in it? What does it do is that it uptakes the nitrogen and it enters the roots of this leguminous plants, and in this way this cycle is completed, and the nitrogen that is usually not uptaken so easily by the plants and organisms, the Rhizobium bacteria helps the plants to take this up and make the soil more fertile, and it makes the whole of this process very helpful actually, because more the fertility, more the yield of crops, and more the yield of crops, the better the production. So all this is interconnected to each other. The other example of the bacteria is also the Azidobacter bacteria, and this is a kind of bacteria that is present in the soil, and this one also helps in actually nitrogen fixation process only. So it does the same functions of making the soil very fertile and useful, and it makes it very aerated. That means it provides the essential nutrients and ions to it with this. So all this is actually interconnected, and we also know that these as Rhizobium bacteria and Azidobacter are not the only bacteria that are present in soil. There are also a number of bacteria that are present in soil that enhance the texture, the quality, the fertility of the soil. And also the texture is improvised when the capacity of oxygen to be taken in and the water to be taken in is increased. So these bacteria, in some or the other way, they help in this process. Now I told you that we are a mutualistic organism between the bacteria and the humans. So what are these? How do we show mutualism in this? Because bacteria are dependent on soil, and they in return make it fertile and nourishing for us, for our use, because of which we get crops, we get yields, we get higher number and good quality products. So in this way, none of the organisms are harmed and mutualism is shown. So I remember telling you this, that we are dependent on to each other and help out each other with the other important processes. While these are certain good bacteria, good microbes, there are certain microbes in bacteria also that are not that good, that are pathogenic in nature. So the example for that is the Staphylococcus.
No more spending your energy and money on coaching classes, or missing classes for rain, for rally, and any other nonsense. Subjects covered by multiple teachers with repeat telecasts. Doubts, concepts, applications, no problem. All explained through great and significant animation. So sit back comfortably in your home and watch Study Spectrum TV channel.
I remember telling you about pathogenic type of microorganisms and beneficial type of microorganisms. Now we've learned the two beneficial type of microorganisms. Ever since we learned that these two are nitrogen fixing organisms, and they fix nitrogen in the soil and the roots of the leguminous plants, making the soil more fertile and plants more rich in the content of protein and other macronutrients that we have required for our body. And the fertility of soil would in return help in increasing the yield of the soil and the quality of the soil. So let's just shift on to the pathogenic type of bacteria. Now in the pathogenic type of bacteria, the first one we had is the Staphylococcus bacteria. The Staphylococcus bacteria's shape is nothing but that of coccus that I explained to you right over here. Now either they can be present in the clustered form, that is right over here, and that is clumped together and clumped together, or in the form of chains of coccus over here. present in the form of chains right from the start till the end. So let's just shift on to our concentration on how this is really pathogenic or poisonous. So what does this Staphylococcus bacteria do? It actually produces a very poisonous toxin like enterotoxin, and it releases it in the food. Now when this enterotoxin, that is a poisonous substance or poisonous chemical that is produced in the cell body of the bacteria, is introduced in the food material, it would do nothing but infect the food. And if this infected food is consumed by a healthy human being or a healthy host, it would finally lead to the occurrence of a disease in that host. And that disease would be nothing like stomach upsets or food poisoning or diarrhea or vomiting. These are the common outcomes that are basically seen after the infections of bacteria. So this would lead to an aggravated situation if the treatment and precautions are not taken up for this on the right time at the right pace. So to avoid such situations, even if the outbreak of the infection has been seen, it is advised for the host or the ill patient to go and to visit the doctor in these cases, and it is curable with timely treatments and better habits of food and sanitation. And even with the antibiotics and the medicines that doctors prescribe to you, with timely and routinely consumption of it, these diseases like and illnesses like the diarrhea, food poisoning, stomach upsets and vomiting and all can be cured. Well, in the list of the pathogenic bacteria, the other bacteria that's present is the Clostridium. Now, Clostridium is the shape that's of the Vibrio shape. So it is very harmful in the manner because it infects the food that are canned. The canned food like having beans, canned beans and all the chocolates and all the yogurts etc. that are canned, it usually affects that type of food, that's the canned food. So Clostridium affects the canned food, and because it has the ability for anaerobic respiration, that means it does not require oxygen for its growth. It can grow in an environment where there is no oxygen too, and hence it proves to be hazardous to the living beings, and that is why it is always better advised to check the expiry date of the food in order to avoid the circumstances to be exposed to the bacteria like Clostridium. Or it is also advised to keep these foods in a very optimum and a very subtle manner away from the sunlight, because sunlight is also responsible, equally it is after being good to us, it is also responsible for creating certain hazards in the stored foods, and therefore the Clostridium is seen in canned foods. So let us just have a gist about it again. So what is a bacteria? I explained to you that bacteria is a microorganism that is larger than the virus, and though it is larger than the virus, it needs a microscope to be observed under. Now when you observe a microorganism under it, the types and the shapes of microorganisms that you would see would be, the first one is the bacillus. Bacillus is nothing but it is rod shaped, and the example for bacillus is the Rhizobium bacteria right over here. The next one is the coccus. Coccus is the bacteria that is of the spherical shape. Now this spherical shaped coccus can be present in the form of clusters and can be present in the form of chains, while the bacillus can also be present in form of chains of rods. The next one is the Vibrio, that is the slightly curved bacteria in shape, or it is in comma in shape. The other shape of bacteria is the Spirillus, that is of the spiral shape right over here. I explained this to you with the help of a laminator having the files to make, and it has a spring, a spiral spring to pin it in the hole of the file. So that spring that you see, that's exactly the shape over here. The next one is a spirochete, and this is nothing but it is a more curved structure. Then we went to learn about the structure and the morphology of the bacteria. What we learned was that the bacteria has free chromosomes instead of having a nucleus. And the other organelles that are present inside the bacteria were the cell wall, the cell membrane, and the cytoplasm. In the cytoplasm were embedded these free chromosomes. Now, after these free chromosomes, what are these free chromosomes actually? Chromosomes are nothing but when the long strands of DNA combine together and they form a huge strand, and these strands they combine to form a long chromosome, and this chromosome is responsible for giving character to every living organism that is present on this planet. The next point was we learned about certain beneficial bacteria, and we learned about certain pathogenic bacteria. What we learned about the beneficial bacteria was that the rhizobium bacteria is a nitrogen fixing bacteria. Over here, nitrogen fixing bacteria that was found in the roots of legumes. Now, Azotobacter was a second kind of bacteria that also helped in nitrogen fixation in soil. Now, why nitrogen fixation was really important? Nitrogen fixation is important because it increases the fertility of the soil, the quality of the whole of soil, and the yield that is produced. And why is nitrogen fixing important in the roots of legumes? Because it gives it whole nutritional content that is present. The next we learned about was that the Staphylococcus bacteria being a pathogen. Now, the Staphylococcus bacteria is pathogenic in nature because it produces a poisonous substance named enterotoxin that is introduced into the food materials. And when this food material is consumed, the final outcome that is seen is nothing but a disease or acute illness. And these may range from the diarrhea, from vomiting, from the food poisoning. And to cure this, a routinely treatment or routinely consumption of antibiotics that will be prescribed by a doctor, or by avoiding the conditions in which the food is kept out for very long, and every time before your meal heating the food and then consuming it. These are certain preventive measures which would reduce the exposure of Staphylococcus bacteria to the food. Now, the next we learned was about the Clostridium. Clostridium is another pathogenic bacteria that is responsible to cause poisonous substances in canned foods. Now, canned foods, I gave you the examples of beans and of other packed foods that are usually found in the market. So, since I told you that Clostridium is a bacteria that does not require oxygen for its growth, hence it is an anaerobic bacteria, and since it's an anaerobic bacteria, it produces really poisonous substances and it excretes them out of their body, introduces it to the food and makes the food furthermore poisonous, which further consumed by the host, that would be the human being for that matter, would be subjected to diseases and illnesses. And to avoid that, this canned food right over here is supposed to be consumed before the date of expiry and should be stored in a cool temperature and should be avoided to face direct sunlight. The next in the types of microorganisms was the virus. So let us just have a gist about the virus. Over here I have made the structure of virus, and this structure is not really very comforting, I know that, but still, the virus is a microorganism that is so minute that it cannot be seen under a microscope either. So what we need is an electron microscope to understand the shape of this virus and how would it be possibly be affecting the host cell. So we observe the virus under an electron microscope. then the structure of the virus, it does not have any cytoplasm or any organelle in it as such. The whole body of this virus right over here is made up of only proteins. The proteins like DNA and RNA over here. The DNA is a deoxyribonucleic acid and RNA is a ribonucleic acid. I'll repeat again. DNA is the deoxyribonucleic acid and RNA is the ribonucleic acid. Now, in the structure, the genetic material, that is the DNA, or over here, DNA or either RNA is present right in the center of the body. So this white trail, this white strand that you watch over here, that is the DNA or the RNA of the virus that defines its virulence and defines its character. I'll explain to you what was virulence. Virulence is nothing but the amount or the capacity at which a virus can cause the infection to the host. So this is the genetic material and this is the whole structure of the virus that is made up only of proteins. It does not have any organelle to it or any cytoplasm in it, in its structure. So I'll just explain to you about how does this virus actually infect the surface of any other host. This is not there in your textbook, this is just for your additional information. So what happens is, this is the whole body of the virus. It goes and it settles down on its desired or targeted cell to infect the cell. So these extensions that you see, it makes a grip on the whole of the surface of cell with these extensions. So just imagine this place, that this is the virus and this is its head, this is its body and this is its source for attachment. So this part is the head part. This is its body through which once the attachment is made, the genetic material goes down and is introduced into the whole other target. So this long tubular part that you see right over here is the body of the virus, whereas this is the head of the virus. And this is used for the attachment. Its structure is really fascinating, isn't it? So now I'll explain to you about the mechanism, what happens. Now once it has settled down onto the surface of the targeted cell or the targeted host where it wants to infect it, this attachment, this part, this would be somewhat like that, and like that it would be creating a web onto it and it would just sting it and sit down onto the surface. So over here you see it's relaxed, but over here it is compressed because it has made an attachment with the cell surface. So this material, this genetic material right here, will be pushed down into the cytoplasm of the cell, of another cell, the target cell that I was talking about. So this. You don't need to go into detail with this, but this is just something additional that I wanted to explain to you. So now these viruses are responsible for the cause of disease in plants and animals as well. Well, in animals, the virus that causes the disease of AIDS, that is acquired immunodeficiency, is caused by the virus that is HIV virus right over here. Now this virus's full form is human immunodeficiency virus. So I'll just write that down. And in plants, the crown gall disease is the disease that is caused by a virus. So this virus is actually nothing but the Azotobacter virus that sometimes in certain conditions it causes these diseases. But I'll just ask you to make an observation the next time you see a plant. What you need to do is you need to pluck out the plant from its root and just observe whether its root nodules are swollen. If it is swollen in form of a crown, then the plant is supposed to be having the crown gall disease, and that's how the name came, the crown gall disease. So let's just have a whole gist of the whole part of virus that we studied. We studied that viruses are really minute and they cannot be observed under a normal microscope or compound microscope. So to observe them, we need an electron microscope. So I explained to you in the start that electron microscope has magnification power of more than 200 billion times than a compound microscope. So imagine how minute it is. Then we studied about the structure of the virus, that in the structure of the virus there was no cytoplasm and no organelles at all. Instead, the whole of the body was made up of proteins. The body over here, this whole body is made up of proteins and not of any organelle or cytoplasm. This right in between, you see this strand over here, this long strand, that's nothing but the genetic material like DNA or the RNA. So I explained to you also the full form of DNA, that was the deoxyribonucleic acid, and RNA is the ribonucleic acid. Then we learned about how this virus can cause infection. How does it infect the other cell surface? So we learned that this part, that is the hexagonal part, is its head. This long tubular part is its body, and these small hair-like structures that you see down is nothing but the structures that are responsible for causing an attachment. Then we learned that how it could connect and attach itself by compressing this attachment structures and make an attachment. Immediately after the attachment was made, the genetic material that was suspended in here in the head of the virus was pushed down through its body into the cytoplasm of another cell for the infection to be caused. And the type and the virulence of infection is depending upon the strain of the DNA or RNA that is present. Then we learned that the virus is responsible to cause diseases in plants and animals. In animals, it caused AIDS, that is acquired immunodeficiency, that was caused by the virus of HIV, and its full form is human immunoglobulin deficiency virus. So, or the human immunodeficiency virus. So the next we also learned was that it could cause infection in plants, and the infection it could cause was the crown gall disease, in which the roots would be swollen in and form a structure like crown. So that is the name from where it came, the crown gall disease. Now let's move on to the further part with the algae and fungi and understand a little bit more about them.